Hi, my name is Georgia Horn, and I'm the Development Director at the Usher III Initiative. It's a pleasure to be here today speaking with you all. Thank you so much to the Usher Coalition for having us. Usher syndrome type 3 is a rare genetic disorder that leads to progressive deaf blindness. Individuals with Usher III are typically born with normal hearing and vision and develop degenerative auditory dysfunction and retinitis pigmentosa. By age 30 to 50, most Usher III patients are legally blind and profoundly deaf. This video was designed by a medical illustration group called Biotic Art Lab to depict the progressive loss of both senses. The nonprofit Usher III Initiative was founded in 2007 by Cindy Eldon, an Usher III patient, and her father, the late Richard Eldon, shown here on the left, to address the lack of treatment options for the Usher III clinical population. Currently, patients may use hearing aids for mild to severe auditory loss, or for profound loss, undergo bilateral cochlear implantation to artificially restore auditory function. While this surgical intervention can be extremely effective, the procedure is costly and insertion can cause irreversible intracochlear trauma, preventing the use of future molecular or regenerative therapies. There is no treatment for the loss of vision, which continues to progress with age due to the gradual death of photoreceptor cells. Existing options for managing visual loss are restricted to lifestyle modifications and the use of assistive technologies like magnifiers, screen readers, and Braille. At the initiative, our sole mission is to change this paradigm through the development of a therapy to halt or attenuate the sensory loss caused by Usher III. Despite extremely limited resources, and as the only organization in the world solely dedicated to Usher III, the initiative has sponsored cutting-edge research that led to the discovery and optimization of BF844, a small novel molecule therapeutic candidate for the treatment of Usher III. BF844 is designed as a systemic treatment for oral administration, avoiding the need for invasive procedures or interventions focused solely on only one of the sensory losses caused by Usher III. Usher III is caused by mutations in the Clarin1 gene, the most common of which in the United States is called N48K. Our work focuses specifically on this mutation of Clarin1, which leads to the subtype Usher IIIA. The abnormal Clarin1 protein produced by this genetic mutation degrades and fails to perform its function, whereas healthy Clarin1 protein supports normal functioning of retinal and inner ear sensory cells. Without it, those cells die. BF844 protects the mutated protein from degradation, enabling it to support sensory cells and preserve function. The initiative has shown that BF844 has been able to preserve hearing function in an Usher III mouse model. To evaluate efficacy, hearing loss was measured using auditory evoked brain responses in transgenic mice across multiple frequencies. BF844 was administered once daily and 2 regimens were tested. The first examined the impact of BF844 after the onset of hearing loss, and the second examined its impact earlier, before the onset. The results shown in the diagram on the right reflect regimen number 2. Looking at this diagram, in which dB denotes decibel, We can see that when administered early, BF844 significantly increased auditory sensitivity in treated Usher III mice, whereas an untreated Usher III mouse only responded to noise at or around 100 decibels, which is about the sound of a wood shop. Treated Usher III mice continued to respond to noises as low as 30 decibels, approximately the sound level of a whisper. While there was some decline in auditory function of treated mice, as you can see in the diagram at the top two parallel lines, auditory sensitivity remained remarkably close to that of a healthy mouse used as a control. The results of this study were published in the peer-reviewed journal Nature Chemical Biology in 2016. One note, although the absence of a visual phenotype in Usher III animal models has suppressed progress in understanding the retinal pathology associated with Usher III, mutated CLARIN1 is responsible for the loss of both senses in human beings. Given our proof that BF844 rescues mutated CLARIN1 in the ear, treatment with this compound should have the same impact on the eye. Regardless, however, the FDA has confirmed that they will not require proof of concept in vision as a clinical trial prerequisite for BF844. Since we last spoke at the Usher Coalition's conference in 2020, we have accomplished an extraordinary amount. This slide provides a summary of the pathway to phase 1 trials to give you a sense of where we stand. Currently, we are focused on manufacturing material in compliance with good manufacturing practices, or GMP for short, as well as our next regulatory hurdle, composing and submitting our investigational new drug, or IND, application. I will go into much further detail in the following slide about each of these points. Early in 2021, we received a grant from the Foundation Fighting Blindness and a major contribution from Grosvenor Capital Management that enabled us to accelerate our development program and achieve several critical milestones. This slide illustrates the milestones we have accomplished over the last year, as well as the activities we've planned moving forward. In this slide, the color green indicates a completed activity, yellow indicates an ongoing activity, and orange indicates a planned activity. Our first major milestone was the establishment of a viable method for producing BF844 at scale and in compliance with regulatory standards, so-called non-GMP material, which is denoted here by milestone flag 1. The batch was used to conduct the 4-week required IND enabling studies, a rigorous series of preclinical toxicity tests used to evaluate whether our compound is suitable for safe human administration. The active phase of these studies concluded in December of 2021, and we have received the final reports in May of this year, as indicated by milestone flag 3. While our analysis remains ongoing, results are very promising thus far and support continued development. With the support of the Foundation Fighting Blindness and the tireless efforts of our team, we also completed our first regulatory milestone in 2021, our pre-investigational new drug, or PIND, meeting with the FDA, milestone flag 2. Held prior to the start of our toxicity studies, feedback from this meeting informed the scope of our safety studies and provided confidence in our approach, as well as confirmation that a healthy volunteer study is acceptable for phase 1 to establish safety and tolerability margins and a PK profile of the drug in humans. Our next regulatory requirement is the IND application mentioned previously. This critical document, which contains all available information on the drug itself, a summary of all the research, including animal testing completed on the drug to date, the design of the proposed human trials, as well as the manufacturing process to ensure the quality of the material produced. It will be used to determine whether or not we're approved for clinical trials by the FDA. We are aiming to submit this application during Q1 2023, as indicated by milestone flag 4. Looking ahead, we are working to continue manufacturing the compound in preparation for clinical trials. We hope to finish production and formulation during Q1 2023, as indicated by milestone flags 5 and 6. With sufficient funding, we hope to commence phase 1 first-in-human trials by spring 2023 in healthy adult volunteers. One note, we have chosen to use healthy volunteers because the overall aim of phase 1 trials is to understand the drug's behavior in humans, not its therapeutic efficacy. In order to do this, one needs a large number of subjects without comorbidities or concomitant use of medications. In other words, healthy individuals. Therefore, this first trial cannot be conducted in Usher III patients because of the small size of the patient population, genetically heterogeneous nature of the disease, and wide geographic distribution of patients around the globe. In addition to preparing for phase 1 clinical trials, one of our biggest priorities is the conduct of a retrospective natural history study to optimize the design of phase 2 therapeutic trials. Through this critical study, we will capture data related to the actual total number of Usher III patients, determine eligibility criteria and the ideal age range for intervention, as well as identifying ideal study sites and investigators, gaining a better understanding of disease progression, and defining necessary trial duration and clinical trial endpoints. To this end, we have begun outreach to providers experienced in managing Usher III care and received a $10,000 grant from the Dutch Usher Syndrome Foundation to initiate data collection in 2021. Importantly, we will also be gathering information from patient interviews to gain their perspective on the impact of the disease on their own lives, their minimum expectations of clinical benefit from a new therapy, and the risks they would be willing to accept to achieve those benefits. The value of a natural history study and creation of a comprehensive Usher III patient database and clinical consortium cannot be underestimated. Not only will it benefit our program, but indeed any therapeutic approach to the treatment of this disease. This is where we really need your help and the help of the patient community. We are so grateful to the physicians, scientists, and organizations who have signaled their willingness to support this vital effort, importantly including the Usher Coalition and Foundation Fighting Blindness. If you have Usher III and you have not already done so, we strongly encourage that you register with the Usher Coalition's patient database, the USH Trust, and the FFB's My Retina Tracker. This will help us immensely in expediting getting this treatment to patients. We really do encourage you to help and get involved in our mission of treating Usher III. We have never been so close to achieving our sole mission of providing the Usher III patient population with a first-in-class therapeutic option. Our team thanks you for your time and asks that you please feel free to reach out to us at info@usher3.org with any questions or concerns. Thank you.